All right. Hello, everyone, and thank you for being here. This is the final and third installment, for now at least, of the Beyond Mammals Exploring Cancer Model series, with this webinar being focused on the zebrafish. I'm Justin Benavides, a program director at NCI's Division of Cancer Biology. A link to live captions can be found in the chat. We have a full agenda today, but first, some quick housekeeping. We will open this webinar with 3 talks from our talented speakers. Please hold your verbal questions during this part of the webinar. If you'd like, you can enter questions into the chat box and we'll get to those following the talks. Once our speakers are finished, we'll begin a discussion, and I'll open the discussion with a couple of standard questions and then open the floor to the rest of the audience. During this discussion, feel free to put your questions in the chat or raise your hand so we can unmute you for a verbal question. Please take care and help us create an atmosphere of mutual respect and sensitivity during this discussion. Finally, I'll say a few words about this series. Here at the Division of Cancer Biology, we are interested in the landscape of cancer models. Much of the research funded by our division is in mammalian models that we all know and love, like the mouse, and a growing proportion is dedicated to developing computational models. But the modeling landscape is wider than just those examples, and in this series, we hope to highlight the work of researchers who use non-mammalian animal models, like zebrafish, to answer cancer biology questions and start a conversation about cancer modeling more broadly. So without further ado, I would like to introduce Dr. James Amatruda, professor at Children's Hospital Los Angeles, Dr. David Langenau, professor at Massachusetts General Hospital, and Dr. Kandice Tanner from NCI's very own Laboratory of Cell Biology. So without further ado, go ahead and take it away, Dr. Amatruda.
Looking okay? Well,
looks great.
Okay, thank you, Justin, for the introduction, this wonderful invitation. Great to be here with my colleagues and to see many friends and colleagues online. So in keeping with the spirit of today's talk, I'll try to highlight along the way some of the strengths and weaknesses of the models that I use and look forward to a robust discussion afterwards. And the topic I'd like to talk today is a central focus of my lab in Ewing sarcoma. This is a bone and soft tissue tumor of children, adolescents, and young adults. It's a small round blue cell tumor and can be successfully treated when it's present in a localized form, that is not spread outside the site of the primary tumor, although that does require a heavy dose of chemotherapy, radiation, surgery with a lot of morbidity. And then, unfortunately, when the tumor has recurred or is in the metastatic state, the outcomes are really uniformly poor, and no trial over the past 4 decades has really changed that outcome. Ewing sarcoma is an example of a fusion oncogene sarcoma. That is a disease caused by a chromosomal translocation that generates a chimeric fusion oncogene. In this case, it's a fusion of a member of the FET family with an X family transcription factor. And by far the most common form is this one, the EWSR1 FLY1 gene, EF1. This was discovered in early 1990s by Olivier Delattre, and we know a lot about the biology of this fusion, how it acts on chromatin to create novel enhancers and really rewires the transcriptome of the cell. But unfortunately, this knowledge has not led to a more effective targeted therapy for this disease. And this is really part of a theme of fusion oncogene sarcomas. Diseases similar to Ewing sarcomas or other small round cell sarcomas such as CIC DUX4 or muscle tumors, fusion positive rhabdomyosarcoma, infantile rhabdomyosarcoma of the VGL2 and CoA2 type. And we've studied a number of these in fish models, and there are a few similar themes. So these fusions act as aberrant transcription factors. Unlike fusions that contain kinase domains, they're very, very difficult to target. Typically, the fusion really rewires the transcriptome of the cell in a very significant way. They are required for tumor growth, and as of yet, there are no agents directly targeting these fusions. One of the things that's really limited progress in this field is the absence of a genetic animal model of Ewing sarcoma. It's not for lack of trying. So there are cell lines that are put to good use. There are some PDX models. One issue is that most cell types do not tolerate expression of the fusion. We'll just a dye or a senesce. So even trying to build an in vitro model has been challenging. There's been some work with mesenchymal stem cells, often sort of a poorly defined stem cell type. And there have been many efforts over the years to develop a mouse model, as nicely described in this paper here. And really those have largely failed because of the developmental toxicity of the fusion and not really knowing the cell of origin, which has prevented development of an effective model. But we have to believe there is a vulnerable population that occurs during early development because of the occurrence of this tumor in children and adolescents. The other remarkable thing about these types of sarcomas is the relative genetic simplicity. So these are Circos plots from a study from Brian Crumpton and Kim Stegmeyer, showing that in many cases, the only identifiable genetic lesion in the tumor is the translocation itself. There are occasionally other chromosomal gains or losses that can be seen, but compare this to a typical breast cancer in which there's dozens or hundreds of structural rearrangements and somatic mutations. And what this means, on the one hand, is that it really limits the number of potential targets to go after for a targeted therapy. But it also gave us hope that the simplicity of this type of genetic underpinnings of this cancer would allow us to build laboratory models. And so a number of years ago, we took a very straightforward approach. We used transposons to express the human form of EVDS-FLI1 under a constitutively active promoter. And we just asked the question, if we inject this fusion at the single cell stage and allow the construct to incorporate into DNA as the fish develops, we figured that most cell types would actually not tolerate the fusion and would die. But the question is, would any cell type survive and give rise to a tumor? And in fact, that was the case. So the fish developed small round blue cell sarcomas that had a lot of histologic and transcriptomic similarity to human Ewing sarcomas. But there were some real problems with this model. It was difficult to work with. There was a lot of developmental toxicity. The penetrance was very low. We deleted P53 from the background, and this led to higher penetrance model, but that's not very physiologic for human Ewing sarcoma. And at the time, we didn't have a fluorescent marker, so we had a lot of work to do to improve the model. And really, it was Elena Vasilyeva, a postdoc in my lab, who took a much better approach in which she developed a conditional model with a lock-stop-locks fluorophore under a ubiquitous promoter that in a Cree-inducible fashion could lead to the expression of flag-tagged human EWS-FLI1 linked to a GFP by a viral 2A marker. And this was important because we wanted to promote the expression of separate GFP and fusion oncogene proteins so as not to disrupt the transcriptomic effects of EWS-FLI1. And under this model, which we believe allowed just a slight delay in the onset of a fusion of the expression of the fusion when we injected the construct along with Cree mRNA, we got a very nicely penetrant model in which we could see the early survival of GFP positive cells that developed into tumors. And the tumors were in interesting locations, typically on the dorsal surface, occasionally on the ventral, often associated with the fins, which are the zebrafish equivalent of the fish limbs. So some interesting anatomic similarity to human Ewing sarcoma. And in work I don't have time to talk about today, Elena went on to show that through this model that there was an previously unknown role for proteoglycan metabolism as a signaling growth hub driving a progression of Ewing sarcoma tumors, and that this pathway could be targeted by small molecule heparan sulfate proteoglycan inhibitors, leading to shrinkage of tumors in vitro and in vivo. And Elena is further pursuing this work as part of a K99R00 award from the NCI. But this really left a big open question, which was, what is the cell of origin of Ewing sarcoma, which was the original reason we adopted the fish to look at this. And there have been many proposed models since the description of this disease by James Ewing more than 100 years ago. The tumor cells have many features of sort of mesenchymal, mesodermal cells, so it's logical to think of there's a mesenchymal progenitor. However, they also have features of neurons, hematopoietic cells, endothelial cells, perivascular cells. So it's really remained a mystery. And in our case, we decided to go back and look more carefully at this mosaic model, again, in which we introduced Cree-inducible expression of the fusion protein into individual cells. And we looked at the embryos and asked, what are the surviving cells? So this is what it looks like when you just express GFP widely in a zebrafish. It's a perfectly happy, normal looking fish, lots of GFP expression throughout the body. In this model that incorporates the EWS-Fly1 oncogene, after a couple of days of life, you can see there's a much more limited population of surviving cells. And this is a typical pattern in which the surviving cells were clustered here at the dorsal surface, with some down here at the ventral part of the embryo. And looking at this pattern, it reminded us a lot of the neural crest. So neural crest is a specific embryonic cell population that arises during early development and gives rise to a number of derivatives, a little bit different in the cranial neural crest than the trunk. And at baseline, the neural crest gives rise to neurons, glia, and pigment, along with a few other specialized derivatives. So the question was, is this a possible cell lineage of origin for Ewing sarcoma? And so we used our model to look into this question, first by doing sort of gain and loss of function experiments. So first off, we asked whether these surviving cells are in fact a neural crest lineage. So Lena made transient SOX10 neural crest BFP positive cells. You can see that they largely overlap with the remaining GFP positive cells a couple of days after expressing EWS-Fly1. More directly, she used a SOX10 CRE in order to activate expression of this EWS-Fly1 from this switch construct only in neural crest. And again, you can see a very similar pattern where it's the neural crest positive cells are able to tolerate the fusion and survive. As a loss of function experiment, she used FOXD3 and TFAP2A morpholinos to recapitulate this mutations of these two genes, which abrogate neural crest development. And one can see that in the absence of neural crest shown here, there's really no GFP positive surviving cells. So the most direct test, of course, was to make a stable transgenic using a mouse-specific form of the SOX10 promoter. We found this to be very specific for neural crested zebrafish, which when crossed with a stable UBI switch EWS-Fly1 line, led to the generation of very highly penetrant lesions, small round blue cell sarcomas, and we did a lot of work to validate that these are very similar to human Ewing sarcomas in their marker expression and transcriptome. Really, the question was, first, is this a mesenchymal tumor arising from a neuronal lineage, and how does that work? So these are single cell attacks, sRNA-seq data from adult tumors showing that this mesenchymal population that arises from the normal zebrafish at this developmental stage really clusters quite nicely with the tumor. So as human Ewing sarcomas do, these tumors exhibit a lot of features of mesenchyme. When we went back to look earlier, we were very interested in the initiating events, something we've never been able to look at because we've never had a genetic animal model. So we looked at 7 days after its induction of the fusion, in which one can see these sort of incipient tumors on the dorsal surface of the fish. We dissected these trunks and compared them to a control, which was just expressing GFP. And right away, one can see that there's very nice separation of these clusters on single cell RNA-seq. And when we looked more closely, we asked specifically, these neural crest precursors are expected to develop into glia, neurons, and eventually into pigment cells. And in the control, that was certainly the case, expressing all these glial and neuronal markers. But in contrast, the tumor cells shown here in this cluster were much more likely to turn on mesenchymal and mesodermal genes such as TWIST1A and T-box transcription factors. So it seemed like somehow this normal neural crest developmental pattern in which these neural crest stem cells give rise to glial cells or to neural progenitors and ultimately to mature neurons was being disrupted. We wanted to know how this worked. So again, in work I don't have time to show today, we did a lot of motif analysis to show that the fusion co-ops developmentally open mesodermal enhancers at family binding sites to really rewire the transcriptome. And showing here that this mesodermal feature of the tumors continues. So this is in situ hybridization for TBXTA, showing that as the tumors develop, they maintain that mesodermal structure. And in particular, we were really struck by an interesting phenotype. So this is where the model can open up new ideas. So these fish have a very strange phenotype in that they develop extra limbs, extra fins. At the base of each of these fins, one can see a cluster of GFP-positive tumor cells. So this made us look at the pattern of limb development and how it might overlap with the pattern of tumor development. Limb development is very well characterized in fish and chick and mouse and other vertebrate species, has a similar pattern in which these T-box transcription factors sort of mediate signals from Wnt to set up an FGF signaling loop that drives outgrowth of the developing limb. And that's shown here, a stereotypical pattern for the pectoral fin showing this pattern of core of Hox genes, TPX, and FGF signaling. Interestingly, in the fish tumor lesions, quite apart from any fin development at this stage, this is very early on, we can see a recapitulation of this pattern. So where the EF1 positive cells are, there's a strong upregulation of Hox signaling with surrounding FGF10 and FGF8 signaling that leads to a signaling loop that drives outgrowth of the fin and the tumor itself. And ultimately, what typically happens is the fin will fade away, the tumor will then start invading into the fish. And typically, one would expect to see TPX4 or TPX5 during normal development. In this model, we could see clearly that TPX-TA is ectopically expressed by the tumor cells. So they turn that mesodermal reprogramming into sort of a niche of limb development to grow out development of the fin. So I'm just about at time. I'll stop here and summarize and say that this work is available in a preprint to show the mesodermal reprogramming of the neural crest lineage. And we think that this may represent at least one type of cell of origin of Ewing sarcoma in humans. There's a lot more work to do in this regard. And there may well be other cells of origin for different subtypes of Ewing sarcoma. We have a lot of other interests that I won't have time to talk about today. In particular, we're looking very closely now that we have an immunocompetent animal model at the role of macrophages in Ewing sarcoma tumor development and outgrowth, and I won't share that now. Just to say you'll hear more from Dr. Tanner, I think, about the tumor microenvironment of fish, and say that there's many other fusions that we need to make models for, sometimes single patient level, rare, unique fusions. And I think the fish is beautifully adapted for that. So I want to stop and mostly acknowledge the work of Elena Vasilyeva and other folks at my lab who have driven this forward, many collaborators and generous supporters, and thank you all for your attention.
Thank you, Dr. Amatruda. And if you have any questions that you want him to answer, feel free to put those into the chat and we'll get to those during the discussion. Next up, we have Dr. Langenau.
Great. Can you see that, Justin?
Yep, looks great.
All right. Jim, that was absolutely fantastic. I'm looking forward to hearing more about the models and then also the questions. So I'm Dave Langenau. Today I'll tell you a little bit about our lab's work in using zebrafish for cancer discovery and preclinical work. I have no relevant disclosures at this time. So we all know that mouse has really been at the forefront of being able to engraft human tumors, and they have really become the major driver of identifying new drugs and therapies. And specifically, you can take either a patient's tumor or cell lines and grow them in a NSG mouse, for example, that lacks T, B, or NK cells. And then, as I said, you can treat them with a wide array of different compounds, small molecules, antibodies, et cetera, and look for tumor regression. And so we and others have decided to use zebrafish as a complementary tool because of the scale at which you can do experiments and some of the imaging, which I'll show you today. So there are two flavors of xenografts that are done in zebrafish. My laboratory really focuses in on using adult xenografts where you can engraft large numbers of cells and they really look like a patient tumor. They can engraft long-term, and I'll show you that in a few slides. But others have also used small zebrafish to engraft small numbers of cells and are really good for rapid assays. Either way, in my laboratory, the idea is, can you use these models now to identify new therapies and really use these for preclinical validation and ultimately moving them into translation? So as I said, my laboratory has really taken the approach of making immune deficient zebrafish. We have 2 different flavors of zebrafish, both of which lack T, B, and NK cells. And the real power of this approach is that you can now raise these fish at 37 degrees. You can also engraft tumors into 2 different places. Either you can engraft 1 million cells or up to 1 million cells into the peritoneal cavity, or you can engraft about 50 to 100,000 tumor cells into the eye musculature. And these are really great for imaging, as I'll show you. One of the other powers of this approach is that you can use oral gavage to give clinically relevant dosing for these animals. And then they are also fairly large scale because, even raising an adult fish to, only costs a few pennies. And in my laboratory, we have about 25,000 animals at any given time. Okay, so now that I've described the models, if very briefly, the first iteration of these came out in 2019, where we identified that there was a drug combination called olaparib and temozolomide. Olaparib is a PARP inhibitor and temozolomide is a DNA damaging agent. And as you can see at the bottom of this slide, when you treat these animals with the combination therapy, the tumors really had a major effect and they went away over time. You can look at these also in real time using fluorescent imaging shown here on the right for various models where you can actually look at more traditional waterfall plots where you can look at individual animals. And hopefully you can see that in a subset of those animals, they really very nicely kept the tumors at bay. And so these were longer term experiments that were carried out over 21 days. And so really very nice, robust responses. And this actually led to a phase 1 clinical trial where we were able to convert a trial for Ewing sarcoma into a basket trial and allowed us to see some patients that had rhabdomyosarcoma being treated with this therapy. So although that's great, we know that in other models, olaparib and temozolomide can lead to therapy resistance. And in fact, that's exactly what we also saw in the clinical trial, especially for the Ewing sarcoma patients, that they could actually progress on therapy quite often. And so we modeled this in the laboratory using mice in this example, where we could see, as we showed in the original paper, that there's really, really nice responses in a subset of the tumors, but in a subset of them, they eventually go on to progress on therapy. So we then took these individual tumors and analyzed them to identify what are the pathways that are driving this resistant mechanism. And because I really wanted to keep this focused in on the models, I'm not going to bore you with all the details of the experiments, but I'll summarize what we found in that about 50% of the tumors actually upregulate the PIK3CA AKT pathway, and this activates a transcription factor called NRF2, which then induces the global expression of ABC transport genes, of which there are 48 different genes. And these ABC drug efflux pumps then allow the cells to pump the drug out of the cells. And so this was really exciting because for years and years, people had been developing ABC transport inhibitors, but because they're such a large class of proteins, they often just upregulate one of the other ABC transporters. But because this was all being directly regulated by the PIK3CA AKT pathway, we reasoned that if you could treat them with an upstream inhibitor, say a PI3 kinase inhibitor, you might be able to suppress this pathway. And that's exactly what we saw in cell culture and worked up that mechanism. But we wanted to see if you could use our zebrafish models to actually dynamically visualize these drug responses in real time. And so in this experiment, we actually labeled the cells with a green fluorescent protein. We also take those cells and we treat them with a CFSE, which is a dye that gets diluted by half every time a cell divides. And then luckily for us, people at Mass General had already made a fluorescent PARP inhibitor, which allows the the drug to be seen in the red fluorescent channel. And so the idea is now you can engraft the tumors into the eye musculature, come in 4 days later, treat them with oral gavage for the drugs, and then witness what's happening in real time. And so what was particularly exciting was that, of course, in the parental cells that are going to eventually be able to be killed by this combination therapy, you can see that their drug really gets into the cells early on. But if you now treat with a resistant model, shown here on the right-hand sides, you can see that they very nicely will pump out the drug, and so they don't have drug inside the cells. But if you now look at the cells that are engrafted and treated with alpelisib, they now become resensitized and can retake up the drug. And this can all be quantified at single cell resolution, shown here on the right-hand side. And as I said, we used a dye that could that is distributed by half every time it dilutes. And so what you can do is you can actually go in and quantify the number of cell divisions that a cell has seen. And so in these experiments, what you can nicely see is that in the control models that are very responsive to therapy, that this is a cytostatic effect, that the cells do see drug and they eventually become arrested and then die. And you can see in the case of resistant models, they really blow through this therapy, and you have a lot of these cells that have rapidly divided and divided up to 5 times, for example, shown here in white. And then, of course, if you treat with the alpelisib drug, you can resensitize them to this cytostatic effect. So a really powerful way to directly image what's happening to these cells in real time and at single cell resolution within the animals. We can also do these same types of experiments in grafting the cells into the peritoneal cavity, and this is just to show some of that data. Sadly, I think we've cropped off some of the images on the RD on the top, but you'll have to take my word for it. Those green bars in the waterfall plots go down further. But it's really nice. This is something that you would do in a mouse, and you can do this at a large scale, hundreds of animals by one individual over time. So, of course, we've also done the very highest bar experiments where you've taken these tumors and engrafted them into mouse PDX models. And again, the same type of thing happens, that you can resensitize these resistant models to olaparib and temozolomide. And this is very nicely tolerated by the animals. And so I think this really has allowed us to be able to identify a resistant mechanism and might suggest the opportunity to start thinking about treating alpelisib with chemotherapy. Probably unlikely to go forward with olaparib and temozolomide, but might be really a powerful opportunity for VAC or other types of salvage therapies in this tumor. Okay, so hopefully I've given you a flavor of how we use the zebrafish for looking at drug synergies and small molecules. What I wanted to do next is tell you about a discovery we made that is using a brand new type of approach to deliver, in this case, not drugs, but a peptide to cells. So this is work that was published, I believe, in 2022, where we have an antibody that is targeted to a cancer cell. And instead of delivering a drug conjugate onto this antibody, we've actually put on a CMV epitope. And this CMV epitope is adjacent a protease cleavage site that is found in the tumor. And so what happens is when this peptide gets to the tumor, it cleaves off this viral epitope, dangles it and drips it back into the cells that are there, and it gets expressed on the MHC class 1 within the tumors, convincing it that it's actually been, or making the immune system believe that it's been virally infected. And so this is really a powerful approach innovated by a collaborator, Mark Cobalt, when he was here at Mass General. And so we did some experiments for him showing that this approach was viable and actually worked beautifully. So here we can take tumor cells where we inject them into the eye musculature. In this case, we're going to use a fluorescent reporter. The M-cherry positive cells will not have the epitope, and green cells will have the epitope. We then engraft those cells, come back 6 days later, inject them with CMV primed CD8 positive T cells. These are all human components. We've used that CFSE blue to dye those cells, and then we treat them with that antibody that can either deliver the CMV epitope or not. And so I won't go through all the details, but I'll show you in a movie what this looks like. So here's the control experiments where we're going to fly into the tumor. You can see equal numbers of green and red cells, and the blue cells, which are not targeted, don't interact or interact very infrequently with the tumors. Now, that's in comparison to a correctly targeted moiety where you can see that we've really nicely got rid of most of those green cells with the epitope, and the remaining T cells that are there really are exquisitely interacting with the remaining tumor cells and presumably getting them ready to be killed. And so this is really another powerful approach. You can quantify all of this at single cell resolution and actually dynamically visualize this in real time. So finally, I'll leave you with one other opportunity that I think we have in the zebrafish. One thing that is very difficult to do in mice is to treat with radiation dosing because the mice are very, very similar to humans, where 2 gray radiation is the maximum dose that you can give to a mouse, and it needs to be given over time. And that's very similar to what we see in the clinic. But what's really remarkable about the zebrafish is that they actually are very, very resistant to radiation. And in fact, you can give them 10 gray radiation doses in one of our backgrounds that lack only T cells using the RAG2 deficient animals, and they are completely alive. If you took a mouse or a human and hit them with 10 gray radiation, they would universally die. And so I think this really allows us this amazing opportunity to think about using them in radiation therapy responsiveness. And I won't take you through these experiments, but we're now working this up in the context of PARP inhibitors and thinking about this approach. So what I hope I've shown you today is that the adult zebrafish models are at least an interesting opportunity for us to think about complexing new small molecules, new types of therapies like these APEX, and even thinking beyond that with radiation. We've used these in the context of CAR-Ts, BiTEs, and other types of small molecules that I didn't have time to take you through today. So with that, I want to say thank you to my laboratory, those people in blue, Yan Chuan, Chi-Chi Yang, Yu-Yang Wang, and Stephanie Strom, for example, did most of the work shown here. Our collaborators, and of course, I'd like to really thank NCI, especially for the R24, which has really been a valuable grant mechanism for us to develop and to distribute these animals to the community. Thank you, and I look forward to questions later on.
Thank you, Dr. Langenau. Feel free to put your questions for Dr. Langenau in the chat. And I would also encourage you to read the chat. Dr. Amatruda is answering some other questions in there. And next we have Dr. Tanner. So go ahead and take it away.
So Justin, can you hear me correctly? Is it okay?
Sounds great. Looks great.
I just want to echo my fellow speakers in terms of thanking Justin. This is just fantastic to feature this model. And for the next 15 minutes, I'm going to talk to you about how we use this as a preclinical model of metastasis. So I always like to start by defining metastasis. Oops. Maybe not. Okay. So in this cartoon, metastasis simply describes a process by which cancer cells leave a primary site, enter into conduits such as lymphatics, blood vessels, survive, exit, and be able to form new lesions in secondary sites. In terms of clinical practice, treating patients with metastatic disease remains a clinical challenge. So how do we then provide druggable interventions simply from discoveries from basic science? So understanding how cancer cells arise due to genetic drivers, due to what molecular subtypes may be distinguishable from that of normal cells, has resulted in targeted therapies, not only focusing on tumors themselves, but microenvironmental factors such as immune cells have resulted in targeted therapies against mounting anti-tumor killing potentials via immunotherapies. And by understanding metabolic differences as well as biophysical differences between cancer cells and normal counterparts has resulted in diagnostic techniques. So these discoveries are rooted in what I would say really basic science. And traditionally, we think about these models as mouse models, as discussed by our previous speakers, organoid models. And I want to introduce today that larval zebrafish has already been used in similar contexts in being able to drive these discoveries to clinical applications. So if we focus on how do we then stop tumor growths, in Rita Fier's lab, what she did was a fantastic study. I just love this work in the sense that she was able to access patients at the time of surgery, take some of their samples, put them back into zebrafish, and zebrafish received the same treatment options as that of the patients, so personalized medicine. And remarkably, the treatment results that were observed in the patients were also observed in zebrafish. And building on these studies, her group and others have recently showed that this has entered into clinical trials. So these are two studies that were presented this year at ASCO, which is like the, I say, forum for discoveries in terms of clinical therapies. But there's one more thing that I need to tell you about metastasis in terms of complications that need to be addressed in terms of designing new therapies, in that the sites at which these cancers originate often dictate the secondary sites where we see these outgrowths. So I'm showing you here for a few subtypes, and all of these, quite frankly, have, I would say, zebrafish counterparts. And these arrows simply talk about latencies, and one can see melanoma is fairly promiscuous, where some of the subtypes have either different latencies or different sites. So the question then is this aspect of human disease, this non-random selectivity, also observed in zebrafish? So in my lab, what we did is we took cell lines that target in mice, either the brain or bone marrow, and simply asked that question if it would also target, when introduced into the zebrafish, larval zebrafish, if they would also home to the brain or the bone marrow niche equivalent of the zebrafish. And suffice to say, what we did is 5 days post-injection, indeed, we observed that cells that home to the brain of the mice also home to the brain of the zebrafish, did not colonize or proliferate within the bone marrow niche, whereas cells that home to the bone marrow also colonized the bone marrow niche in the fish, and we didn't see much of a colonization in the brain. So this microenvironmental fidelity is conserved such that there's sufficient conservation between what we define as these microenvironmental niches to observe what is seen in human patients. So for the rest of the talk, I'm going to talk about how could we then use our zebrafish model to talk about different areas of exploration to identify interventions. So I mentioned that we can use this model to address how tumors grow. More importantly, because of the ability to visualize, we can target then how do they escape? Could we understand why the tumors spread? How are they surviving within these conduits? These are harsh environments. And finally, once they get out, what allows them to adapt to these new organs that have distinct microenvironments from their origin? So for the rest of the talk, I'm going to talk about how we could use zebrafish in terms of bringing new insights, in terms of non-tumor interventions, focusing on the tumor microenvironment. And more importantly, understanding in terms of differentiating what may be some biomarkers as akin to metabolic mapping to be able to add some insight in terms of therapeutics. So in this movie I'm showing you here, this is a forebrain, midbrain, hindbrain. So this is a 3-day-old fish. The blood vessels are labeled in gray. A type of immune system, neutrophils, immune cells, neutrophils are labeled. And we introduce cancer cells at day 2 days post-fertilization. And in this inset, you see that we're able to recreate what I've shown you in that schematic, in that cells are able to arrest, they extravasate, and unfortunately, these human cells are able to divide. So this is building on work others have looked at. I've listed these labs here, the visualization, which is one of the powerful tools that zebrafish presents to us. But I'd like to focus on what types of insights we get from looking at these tumor-stromal interactions. So I'm going to highlight 3 studies that were simply talking about what insights we got from simply visualizing these processes. So Clancy's group showed phenomenally that once you introduce human cancer cells, that they show this preferential colonization through these endothelial hotspots, indicating that not only do the tumor cells are an important target, but what types of endothelial interfaces, and could we talk about using that as a therapeutic interactions? What Jackie Goat showed very phenomenally is not only within these conduits, the migration is influenced by these hemodynamic forces. By being able to quantify how much force that these tumor cells showed a differential extravasation, meaning the ability to leave their primary site based on what types of forces they believe, introducing that biophysical parameters within the microenvironment are important. In my own group, what we've done is by using techniques repurposed for single molecule biophysics using optical tweezers, we've shown that not only are the cell's biophysical property important, but the environment in terms of what types of mechanical barriers these cells face also influence the cell's ability to colonize one organ versus the other. Here I'm just showing you a slew of model systems, zebrafish, microfluidics, and again, using optical tweezers to trap zebrafish to quantify these forces directly in the living animal. But for the rest of the talk, I'd like to go back to, juvenile and adult fish. The larval fish are phenomenal in terms of the ability to have optical accessibility. They present the ability to look at multiple organs, albeit in a system where only an innate immune system is present. And as Dave already mentioned, that we'd like to be able to scale this to adult or juvenile zebrafish, where we have the capabilities to look at later stages of metastatic outgrowth and to building components of immune systems, namely adaptive immunity, for GEM models. So this, where I'm going to talk about a model that was developed in Len Zon's group by Elizabeth Pathan, and some of the work that has been extended to Richard White and his group and his cell lines. So we love to visualize, and I just want to really go back in terms of, should we also understand that not only druggable targets being the tumor cells themselves, but the microenvironment, and illustrated in this micrograph. These are now fish, adult juvenile fish, where on the control, this is simply a cubic, so a cleared brain, and we could appreciate the blood vessel architecture on the left in a normal brain. And on your right, if one thing you can compare as a function of just by eye, you could see that the vascular system is clearly messed up when a tumor is present. If we zoom in, we can really appreciate the changes that are seen as a function of tumors versus. So even if we eradicate this tumor, the reality is we still also need to heal the microenvironment because that in itself has been disrupted. So can we then understand how these factors are changed, and more importantly, also understand how our therapeutics will go in terms of healing the organ as a whole? So for us to be able to bring that level of single cell imaging that is beautifully done in the larval fish that we and others have shown, the ability to be able to immobilize the fish safely and do longitudinal imaging over several days required a bit of a few tricks. So on your left, what we've developed here, the intramural program, is that we place these fish in 3D bioprinted materials. This is a tube now that can be used to offer drugs such as oral gavage, that's shown on your right. But more importantly, what you're not seeing is these external devices that allow the fish to think it's swimming because we flow water across its gill. In this system, it allows us to do longitudinal imaging that traditionally has not been shown to be easily done in zebrafish. So here I'm showing you that we're able to do oral gavage. We can follow now drugs and follow these fish as a function of drug treatment. But today I want to focus on what can we see. So on your left here, again, going back to this melanoma model where we have the ability to introduce zebrafish melanomas carrying the human BRAF mutation that's commonly found in zebrafish developed by Richard White and Len Zon's group. And what we've seen here is that we've introduced and we can have brain metastases and skeletal metastases all within the same fish, allowing us to have access to multiple organs and do with near simultaneity imaging of these two sites. On your left versus your right, what you're looking at is simply T cell dynamics between these tumors. Melanomas are blue, T cells are labeled in magenta. And you could appreciate that there's a difference in terms of the accumulation. There's a difference in terms of the motility that's observed as a function of these two sites. Of course, because of the beautiful transgenics of these animals, we could also look at other immune systems. So what we'd like to be able to do is translate the ability to visualize these differences in immune infiltrations, motilities, into functional and therefore appreciation for therapeutic interventions. So, of course, one can do single cell transcriptomic analysis. We could identify these functions, but that does not allow us in part to understand some aspects of why these cells behave in this way in a given organ microenvironment. So today I'm going to introduce how we could then add in multiplex measurements where we do fluorescence lifetime imaging microscopy, which allows us to optically detect in a living animal with single cell resolution what type of metabolic behavior that these cells are exhibiting. So it goes as this. For those of you who may be familiar, this is work by my PhD advisor, Enrico Gotton, Melissa Scala, and so forth, in terms of being film pioneers. And all it does is, instead of looking at the intensity as shown here in this image of the cells themselves or whatever organ of interest, but the time that these cells, this excited state stays, let me say this again, the time that the once light excites these fluorophores, the time that it takes before it gets down to the ground state is known as a lifetime. And that could then be used to be able to molecularly fingerprint the types of species that are present. In this cartoon, what we're looking at is free and bound forms of an enzyme known as NADH. And what we can then use by looking at these metrics, what are the relative contributions of oxidative versus glycolysis versus oxidative phosphorylation. So how does it look like? You have these heat maps. Here, this is now in terms of nanoseconds. So this is not fluorescence intensity. This is fluorescence lifetime. And you can see that the brain metastases are fairly more heterogeneous than that of skeletal metastases in this issue. But then we translate this into relative contributions of glycolysis versus oxidative phosphorylation. And on average, what we observe is that the brain shows more glycolysis than that of the skeletal metastases. But the power of this technique is that not only can we look ensemble, but we could identify with single cell resolution. And just as a validation, our ex vivo models validate what we're seeing from these in vivo measurements, is that we can look now at individual T cell metabolomics in one fell swoop. So I'm focusing here on normal versus brain metastases. And one thing you could take away from this by looking at these heat maps, looking at the fluorescent lifetime profiles, is that there is a difference in terms of the heterogeneities that we could quantify here. On average, what we see is that the T cells that are found in the normal adjacent brain tissue show a higher rate of glycolysis compared to that of the T cells found within the brain metastases. Now, if we get into now individual profiling, what we observe is that there's a tighter distribution of these cells within the normal adjacent tissue compared to this broad distribution, simply meaning that the T cells within these brain metastatic lesions can occupy different states. Some of them are more glycolytic, some of them are less glycolytic. So I hope in terms of showing you these different modelings, different imaging modalities, that we could begin to use the Zebrafish model to look at other types of factors that drive, I would say, therapeutic efficacy in terms of looking at the organ microenvironment and organ-specific microenvironment in the case of metastatic melanomas. In the case of diagnostics, PET imaging is already shown in vivo to be able to distinguish tumors from normal tissues. But here, in terms of providing a molecular profiling, that we can monitor to understand how then the environment responds to therapeutic interventions. I made a case to show you that this has already been done in the clinic for larval zebrafish. Dave has already talked about what has been done in the adult and juvenile fish. And for me, I make a case that these metabolic or multiplex imaging can also be useful in terms of understanding the mechanisms of why some tumors respond versus others. So I end with this slide in the sense that when we talk about this pipeline from going from basic science discoveries to the ability to have clinical applications, we need to expand this landscape, not only to involve other animal models, but adult and larval forms of zebrafish allow us to bring some additional mechanistic insight that may be underappreciated. So with that, I'd like to thank the lab. I'd also like to thank the zebrafish community, both intramural and extramural, basically giving a shout out to Ramon Sood, Kevin Bishop, and Blake Carrington over in NHGRI, who have been long-standing collaborations, and of course, my funding sources. And I'm thankful for any questions that you may have.
Thank you, Dr. Tanner. So we're going to go ahead and move right into the discussion at this point. But feel free to put any questions for Dr. Tanner in the chat, and I'll be sure to get to those following our standard questions here. So first, thanks all of you for excellent talks. I'm just going to get the discussion started with some questions that I've been asking to all of the webinar speakers for the past couple of installments, just to get a baseline for zebrafish as a model. And I know that you've answered some of these questions either in the chat or in your talks, but really just to hammer home the strengths and weaknesses of the zebrafish. So first question is, what is unique about zebrafish that makes it useful as a cancer model? And I'll ask James to answer this one first, since you were the first speaker, and then we can go from
thanks. Could go on for a long time, but I'll say a couple things. It's a combination of sort of speed, low cost, flexibility. So in our case, we were able to generate large numbers of fish with kind of a Hail Mary experiment, express a transposon-based construct widely and ask, does something come out the other end? It'd be difficult to justify doing that with a mouse, but that gave us the proof of principle and actually the first clues to make a more refined model. Second, it's, as you've seen very beautifully in the other talks today, imaging. So, and across multiple scales of development. So single cell level at the embryonic larval stage, all the way up through juveniles and adults, and couple that with drug screens. There are caveats that we could get into with administration of drugs, in the water, IP, gavage, as Kandice showed. And there's, questions about PK and PD that come up always with zebrafish. But I think in our work, we try to focus on the potential advantages and overcome or, work our way around some of the disadvantages.
Dave, you want to go next?
Jim summarized it very well. I would say the only thing I would add would be that in Jim and I've talked a lot about this, that the fish are really good for discovery. And so we think that, you make a discovery in the fish, and then when the next model is required for you to validate, that happens. And so a lot of our work will do something in the fish and then move into the human patient samples or into mouse xenografts, for example.
And Kandice.
So I'm going to add a different lens because, I started off life in doing three-dimensional culture systems, right? So why did I move, some of my lab's work into zebrafish? And the reality is that to build these complex organs are difficult, right? There's heterogeneity in terms of getting the correct fluid forces and so forth that make an organ physiologically resemble what it is in a human is difficult to achieve, and very few labs have the capability of doing so. And I would argue that zebrafish and other types of animal models allow us to understand this complexity with single cell to make our 3D culture models better, right? So in some ways, this system has the natural stoichiometry, has the natural vascular system. The microenvironmental fidelity is phenomenal in terms of the genetics and so forth has been done by developmental biologists. I want to make sure that these people get credit for all of the work that we could trust that when we use our fish systems, it mimics what may be seen in a human system. So to me, it's an underutilized tool in understanding just normal physiology.
Awesome. Thank you. So the second question that we always ask is, what are the most important differences to keep in mind between cancer in humans and cancer in zebrafish when doing these studies? And I think we can do the same order here. So Jim, go ahead.
Okay. One that I think about a lot is the range of, so the spectrum of histologies that can be modeled. So at a simple level, many of us have found that fish is an excellent genetic model for leukemias, as Dave first showed 20-something years ago with T-cell leukemia, for sarcomas, for some brain tumors, liver tumors, skin cancers, melanoma for sure. For carcinoma, so the most common cancers in adults, lung, breast, colon, prostate, the model has been less utilized for primary genetic models. And you could say, well, the fish don't have a prostate or mammary tissue. So, obviously it's a non-starter. But I strongly believe that, aspects of cancer biology, meaning the genetic and epigenetic underpinnings of cell transformation, cell microenvironmental interactions, immune, as Kandice is showing us, that can be studied with the right lens to even take on the challenge of studying carcinoma biology, for example. You just have to be aware that sometimes the direct translation in terms of transcriptional drivers of a particular tumor histology may be quite different.
I would say one of the things that I've always faced with is people will say, well, we've cured lots of mice from our studies that with drugs that have not worked in patients. And so the fish is going to be just very similar, that models are just models. And the best, most informative model is ultimately human. And so what is the bar that's needed to move something from a discovery into the clinic is always, I think, the challenge. And it doesn't make the fish better or worse. It's just that it's something you have to consider whenever you're working with models. That'd be my 2 cents.
So I guess I'll chime in and say, we have to be realistic about what stage of development are we using and what are the caveats, right? And the larval system is great to look at if you want to have innate immune cells present, but you're not going to be able to look at adaptive immune response. But that's comparable to any sort of, I would say, immune compromised mouse, for example, right? But the thing that in my mind requires systematic comparison is simply differences in physiological temperatures, right? Dave has showed that you can do the xenograph studies in the adult animal at 37 degrees Celsius, but is it that if we administer drugs at 28.5 degrees Celsius versus 37 degrees, there's going to be differences in how these drugs react? And in my group, we've tried to do the systematic comparison of what differences really occur and what are we phenocopying and what are we not. And I think moving forward with any model system, we're just going to have to be transparent and say, look, if it is at 28.5 degrees, I'll use the example that we have in our group. We looked at macrophage, I would say, activation. And the difference between culturing human macrophages in the fish at 28.5 degrees versus 37 degrees Celsius is simply that there was a delay in 3 days before they mimicked what they would see at 37 degrees Celsius, right? So we presented that, and we're not the only ones doing this. So it may not be sexy, but, and I hope that NCI, the funding agency, which is not me, appreciates that. But with any model system, this is a side-by-side comparative analysis that is needed for us to really bring to strength, such that we know when we say this, what exactly we're seeing.
Those are great insights. Thank you. And then my final standard question before we get into our audience questions here. What do you all see as the major obstacles to more cancer researchers adopting zebrafish as a model in their labs?
Okay, so I'll start. So one of them is, especially for young investigators starting out, entering a new institution, if there's not already established fish infrastructure, it can be a big lift, even a conceptual or theoretical lift. So someone coming in saying, I need 120 mouse cages, no one would blink an eye. Occasionally we'll find new investigators, I want to work with fish, and it seems like a huge lift. Now, many successful groups have started out very small with a couple of racks, a relatively low footprint, and expanded from there. At centers like the NCI or Mass General or our center, we're blessed with having very big infrastructure. So that's one of the first obstacles. The second is, as Dave and others have pointed out, conceptual, sort of getting people to embrace that the model is relevant. And I think the quality of the work described by my colleagues, they just, makes the case that big insights into biology, tumor origin, behavior, metabolic response, et cetera, can be generated by the model. So sort of convincing the community will come, I think, through doing high quality work. And then there's, it's a wonderful community in terms of sharing reagents, protocols, strains, lines, plasmids. In that sense, more aligned maybe with our Drosophila and C. elegans colleagues. And that's one of the pleasures of working in the field.
I think one thing that's specific to the models that we've developed is there is additional husbandry and other types of things that are not standard in zebrafish labs. So, learning how to raise fish at 37 degrees, realizing that the fish eat double the amount of food because their metabolism is double. All those types of things, figuring out how to image, like Kandice showed us, at 37 degrees and in real time in an adult fish. So there are some additional husbandry types of things that are needed in our models compared to just a standard zebrafish lab. But other people have taken these things up and been able to grow fish in water baths and things like that without a huge system that's dedicated to 37 degrees.
So I think, I would co-sign everything that has already been said and add one caveat. One of the things of being non-traditional is that, how do you break into prime time? Of course, Jim and Dave have done that successfully and so forth, but how do you do that if you come from a non-zebrafish background? And I think it really comes back to saying that there is no competition. There is no competition between any model system, right? We have to appreciate that we're all working towards the same goal, in that we want to bring therapeutic interventions and cure cancers in humans, right? So your model system is not a competitor to this model system, whatever your favorite model system is, right? And just appreciate that each of these systems can bring something to the table, right? And if we could bring that awareness to the field and also be transparent about the limitations, right? I always tell people in my group, we are not going to overpromise and underdeliver. Right? So essentially say what we mean and make sure that what we say, when is it valid, what regime is it valid, and what regime it is not. And that's it.
Okay, thanks to all of the speakers. We have about 10 minutes left to take some questions from the audience. So if you haven't already, feel free to put your question in the chat, or if you want to ask your question verbally, feel free to raise your hand and I can request that you be unmuted to ask your question verbally. First, I see a question in the chat from Dave Builder for Kandice. He says, really interesting talk. Any insight into the difference in TMEs that drives the differential T cell metabolism in the different organs that are metastatic sites?
Hi, Dave. How are you doing? So you're absolutely spot on. And what Norbert Woong Young So and Patricia Gordon has done in the lab is simply dissect using all of these amazing transgenic animals, and in the case of macrophages. So if you have CSF1R mutation, and this is a mutant that was originally developed now I'm blanking on his name. I apologize. I stopped drinking coffee. But anyway, nonetheless, we do see differences in terms of the metabolic profiling of the cells and in terms of antigen presentation. This is something that we've been trying to wrap our heads around. And in fact, it would be great to have your insight because we know you also work with macrophages in the Drosophila, to really show that just by tuning the presence of other microenvironmental macrophages in this case, you can tune the immune metabolic profiling. Okay.
Okay, I'm not seeing anybody adding any additional questions in the chat, but I do have a few questions. I think it was Jim, you mentioned the nuances of different drug delivery methodologies in zebrafish, and you said you didn't want to go into the nitty-gritty, but I am curious about the nitty-gritty. Can you maybe go deeper on those different methodologies and the pros and cons of each of them?
Sure, with the caveat that this is not something I have a lot of expertise or experience with. So Dave would probably be better placed.
Thanks, Jim. I'll take that. Yeah, so traditionally, when you are using small larval zebrafish, the only way to really give them drugs is to deliver it in the water. And so a lot of people will use water delivery, and it's pretty much unknown how much drug is actually getting into the fish. There are some nice new elegant studies coming out that they can take whole embryos and actually look at drugs that are in them, and that may give us some new insights into that. As the fish get older, you can treat them by putting oral gavage, and you can actually deliver a certain amount of drug orally, and that really mimics more, we think, what you would do in patients or in the mouse xenografts. And then, as Jim also alluded to, you can do things like deliver drugs. Most of the time, we do those by peritoneal cavity injections. Remarkably, I don't know why, but they actually will engraft, or they will go into the blood system at that point. And then there are others who have also done delivery to the eye vasculature for different drugs. Jim.
I'll just add that there have been published techniques on gavage of larvae using very fine polished microcapillaries, which is a kind of a bravura technique, but it has been done.
Yeah, good point, Jim. I think most people would say it's tough, so that's true. But most of the studies are delivering them into the water, but you're right. And I'm sure people can micro-inject into the fish larvae as well. It's just not as common, I would say.
And then, sorry, Candice, real quickly, Liz Patton's lab recently published a model where they made food pellets. So incorporating a BRAF inhibitor, I think it was, and the fish go gobble them up. So it's sort of free swimming delivery as you might use with mouse chow.
So I just want to add one of the benefits that I meant to discuss in the talk is that, off-target effects and to understand exactly how much drug is actually getting in there, right? You can do so systemically to see clearance because zebrafish have guts, the liver and so forth, to see if the drug via whatever route of administration you do, how much of it is actually getting to the organs of target. Right. And the zebrafish allows you to do so in a rapid way that you simply can't do in a mouse.
So I'm seeing a question in the chat from Mark. He asks, I think this is mostly for Dr. Amatruda, but feel free to chime in. Is it accurate to say that the reprogramming of neural crest into mesenchymal is adventitious?
Is it advantageous for the tumor?
Yeah, I'm assuming that advantageous is the intention. Yeah.
Yeah. So a wonderful question, and the truth is we don't know. To the extent in which we've tried to go directly to modeling, inducing the expression of the fusion in mesenchymal tissues, it really has not resulted in good tumor models. And one of the big caveats is that the whole concept of what is a mesenchymal stem cell is relatively poorly defined and may be quite different for different tissues at different stages of the development of the organism. So it may be a limitation of the technical approach we're taking. But it is a great, interesting question. One thing we think a lot about is the the tumor cells themselves, if you start with the human tumors and look at them, they really retain this sort of hybrid features of both mesenchymal and neuronal gene expression. And so that may give the cells certain properties in terms of their, metabolism, chemoresistance, interactions with the tumor microenvironment that uniquely allow them to survive as a primary tumor and to grow and to metastasize and adapt to new environments. That's something we're quite interested in, but haven't looked at very intensively yet.
Okay, in the chat, a clarification by adventitious, Mark means happening or carried on according to chance rather than by design or inherent nature.
Great question. It opens a lot of philosophical issues about, what is a cell of origin? I guess I would say we have data showing that in this model, there are specific features of the precursor cells that make them very, well poised to tolerate expression of the fusion. And if we look, later in, for example, in the development of the neural crest, it doesn't give the same phenotype. So there's some nature of those cells, and the fact that the output is so similar to human Ewing sarcoma, I would say it's maybe less about sort of chance in this case. But those are difficult questions to get at because we haven't really tested every single cell type, for example. And in humans, we can't really look back in time and tell what was the cell of origin. Because likely, as with many pediatric cancers, the origins of these tumors occur during embryonic, fetal, or neonatal development.
Okay, I have maybe one more question for myself selfishly before we close. And I just want to encourage all the watchers here to check the chat because Jim and Dave both answered quite a few questions in the chat and replies. But especially in your talk, Kandice, there were so many incredible images, like live images of zebrafish. And there was a little bit of discussion obliquely mentioning that it's more difficult to image in the adult zebrafish than it is in the larval zebrafish. I think it's a little bit intuitive why, but can you maybe speak a little bit more on the advantages and why it's easier to image in larval zebrafish versus adult zebrafish and how you've been able to image in adult zebrafish?
So, I want to just always say is anything is optically accessible. It depends on what type of spatial resolution you need, what type of contrast you need. So I start with that as a trained spectroscopist. Having said that, larval zebrafish are amenable to many types of conventional fluorescence microscopy simply due to the fact of the wonderful transgenics that already exist in terms of labeling different architecture like blood vessels and so forth. So you can use the size and the relative opacity of these tissues allow you to be able to have single subcellular resolution using standard conventional microscopy techniques. And you could also couple them using mechanical probing. We've used optical tweezers in them. Melissa Scala and others have used film microscopy. You can do the metabolic profiling. And due to the fact that you could immobilize these fish in these very weak, dilute agarose gels, right? And the fish can grow and survive due to the fact that at that size, oxygen could diffuse in nicely. But fish remain fish and they need to have, as they mature with gills, that water needs to be able to be flown over them. That's why they swim to facilitate this oxygen interface. And the issue with imaging fish over long terms is simply how do you keep fish alive, immobilized, with the correct anesthesia, such that you could do this type of longitudinal single cell imaging using the same standard microscopy techniques that I've mentioned. And a few people have come at these tricks in terms of building microfluid devices or being able to immobilize them just briefly for maybe 30 minutes and then be able to recover the fish and move forward. And in our case, what we did is simply relied on simple 3D bioprinting, and then by external hydraulics control the flow over these gills to make it seem as if the fish are swimming. Right. So once you have that in place, and again, this is due to the fact of we have bioengineers here at the intramural program as well, right? And just by these sort of like external tweaks, we could then bring the same sort of optical accessibility that has been done with larval zebrafish to the adult fish. Now, how easy is this to implement, right? I talk about it as if it was a trivial matter, but the reality is that bringing people who have this type of background to people who want to do these types of imaging is also sort of like a barrier, right? I straddle both fields, but the reality is that there are others who can do this in a way that's much more intuitive and so forth. And we just need to bring those types of people into the community. I hope I wasn't too long-winded, but that's essentially what I think is the major barrier. Simply, how do you keep the fish alive, right?
No, I'm also super interested in imaging technology, so I appreciate the discussion. But that does bring us to time. So thank you, audience, for the great questions, and of course, all of our speakers. The talks were incredible. I hope you learned a little bit about cancer modeling today. Be on the lookout for an evaluation survey in your inbox following this webinar. We'd love to get your feedback on this series. This is the final one for now. Recordings for all 3 webinars in the series will be available on the DCB events website. So that link will also be in the email that I send later to all of you. I hope everyone has a great afternoon. See ya.
Want to
Thank you.
stay on, Justin, or are we done?